Okay, hi everyone. Today we're going to be covering the IGCSE Human Biology past paper. So this is from 2021 November. This is paper 1. You can see that here. And this paper should take you about an hour and 45 minutes to complete. And the total marks for this paper is 90. And so for this paper, it tests you on the entire syllabus. Okay. So let's go over question 1. All right, so the enzyme catalase is found in the liver. Catalase acts as a catalyst, which, when added to hydrogen peroxide solution, causes the solution to break down into water and oxygen. So I'm just going to write out the formula just for your own reference. So hydrogen peroxide is H2O2. So for anyone that's doing chemistry, this might be helpful. So catalase is the enzyme that breaks it down into water and oxygen. So oxygen is a gas, which is why we're going to see how many oxygen bubbles are being produced here. So it says this is a student's method. A student investigates the increase in the rate of breakdown of hydrogen peroxide into water and oxygen using catalase. So this is a student's method. They put 100 grams of liver to 100 cm cube of water and then mix them into a smooth paste. And then they put 10 cm cube of that paste into a beaker at room temperature and then add 1 drop of hydrogen peroxide to the paste. Then they would assess how many oxygen bubbles are produced on a scale of 0 to 5, where 5 represents the most bubbles, whereas 0 represents no bubbles. So these are the different conditions that the student did the investigation in. So it says here, liver paste is heated to 70 degrees. So we know that enzymes are affected by temperature. So enzyme activity is affected by temperature. I'm just going to write a little graph here. So if you had temperature and then you had rate of reaction, as the temperature increases, the rate of reaction increases, and that's because there is more kinetic energy, so therefore more frequent collisions between the substrate and the enzyme. However, at higher temperatures, what happens is that the enzyme changes shape, it becomes denatured, so therefore the active site changes shape, and the substrate can no longer bind to the active site. So then what would happen is this, right? A decrease in rate after the optimum temperature. So the second condition that they also use is it's kept at 3 degrees for an hour. So again, lower temperatures means lower kinetic energy, so the particles are moving much more slowly, so therefore less frequent collisions between the enzyme and the substrate. So they've also used acidified liver paste, and also they've added alkali. So they've changed up the pH there. So if you have, if the enzyme is working at a pH that is not its optimal pH, then the enzyme would denature, and then you would have a decreased rate of reaction there. And then lastly, they have liver paste with no hydrogen peroxide added. So the last condition, which is no hydrogen peroxide added, is basically the control for this experiment. So you want to make sure that any bubbles that are produced is due to the reaction between the catalase and the hydrogen peroxide. So again, the hydrogen peroxide is the substrate, and if you don't have the substrate, then you can't have the reaction. All right, so these are the students' results here in their notebook, and it says, give the students results in a form of a suitable table. Scroll back up. So the student has list conditions that they've done the liver paste experiment in, and the numbers here tell us the oxygen bubbles that are produced according to the scale. And we want to present this table in a way that is easy to digest. So we can have one column be the independent variable, and that's the different conditions. And the second column would be the dependent variable. So these are all the numbers and the scale of oxygen bubbles produced. Now, in order to make it a little bit more digestible, we can sort out these numbers into from lowest to highest or from highest to lowest, just so it's a little bit more easier as you're going through the rows of the different columns. So let's go back here and draw it out. So we have to draw a couple straight lines. Make sure you do this with a ruler. And we need to write conditions in one column. And then also the scale of oxygen bubbles produced in the other column. Produced. So let's go back up. So the highest one should be room temperature and alkali added. So that's 5 and 3. So we have room temperature, and that's 5. And then alkali added is next. Alkali added, and that's 3. And the next part is acid added. So that's 2. So acid added, and we have 2. And the next part is kept at 3 degrees, and that one was 1. And then we had 70 degrees after that, so that would be 0 because it's denatured. Lastly, we have the control, right? So no hydrogen peroxide added. So that would be 0. So state why this investigation is not reliable. So whenever we think about the word reliable, always link it back to repeats. So I'm just going to show a little diagram to show you the difference between reliability and accuracy. So reliability is basically how consistent your results are, whereas accuracy tells you how close it is to the true value. This you would have accurate and reliable, so all the data points are at the bullseye and the data points are close to each other. And the next one here, we have accurate and unreliable. So you can see all the data points are more spread out, so that makes it unreliable. And the third one here, we have inaccurate and reliable. So all the data points are away from the bullseye, but the data points itself are still close to each other. And then lastly, we have inaccurate and unreliable. So how do we remove this? Okay. So it says state why this investigation is not reliable. So when it comes to reliability, we have to link that to repeats. So let's say they did not repeat the experiment. Give a reason why the results of this investigation are not accurate. Well, earlier I said that accuracy is how close the value is to the true value. And we can see here, we're basically measuring the bubbles in terms of a scale from 0 to 5, which is very subjective. So we can write that down there. So the results are based on a scale which, no, not which is, but that is subjective. Okay, so for one person, there might be a lot of bubbles, but for another person, there might not be that much bubbles. It's very subjective. And then part 4, describe how the investigation could be improved so that the conclusions could be more accurate, reliable. See the word reliable, we should always link it back to repeats. Right, and then more accurate. So when it comes to making an experiment more accurate, we need to change up the experimental setup that we're using. So we might change up the apparatus. In order to improve the reliability, the student can repeat the experiment. And in order to make it more accurate, we can, for example, measure the amount of oxygen gas that is produced, and we can use a gas syringe for that. So the student could repeat the experiment So this part isn't technically part of your marking scheme, but I'm just going to write at least 3 times. So at least 3 times to improve reliability. And the student could measure the volume of oxygen gas produced using a gas syringe to improve accuracy. Okay. Part B1. So it says state the condition where the enzymes were the most active. So that would be, I'm just going to highlight this, that would be the room temperature. Right here, room temperature. Okay, state the reason for including the beaker with no hydrogen peroxide. So I mentioned this earlier. So this would be the control setup. So the reason why we have the control, again, is to make sure that any bubbles that are being produced is due to the enzyme reacting with hydrogen peroxide. And then lastly, part 3, explain the result obtained when the conditions were acidic. So if we scroll back up, in acidic conditions, the oxygen bubbles produced is 2 out of 5, which means the rate of reaction has decreased. And I mentioned this earlier, but when an enzyme is away from its optimum pH, it might become denatured. It would become denatured, sorry. So we can say the rate of enzymatic activity because it is away from its optimal pH. Because of that, the enzyme has denatured, which means the active site has changed shape and the substrate won't fit in the active site. Therefore, there are fewer enzyme substrate complexes. So enzyme substrate complexes are what we refer to when the substrate is bound to the active site of the enzyme. Okay, let's go over the marks here. 1 mark for reduced enzymatic activity, another mark for not at its optimal pH. A third mark is active site has changed shape. And then fourth mark is fewer enzyme substrate complexes. So some extra points that I've added here is the term denatured. So using the term, I think it's good practice to include the word denature whenever you're talking about, answering enzyme questions. And also substrate won't fit into the active site because this is also another mark that might be included, like in other questions. Okay, let's move on to question 2. Okay, question 2. Molecules move in and out of the cells by 3 methods. These 3 methods are diffusion, active transport, and osmosis. So part A, it says give a definition of diffusion. So you do actually need to remember the definitions for all three of them. So diffusion is basically the movement of molecules from a region of high concentration to a region of low concentration. Or you can say down a concentration gradient. So an example of that is if I were to spritz perfume into the room, the perfume particles or molecules are not just going to stay in that one spot, they're going to diffuse throughout the room. So that's basically what diffusion is. So it is the movement of particles from, or molecules from high to low concentration. So the 2 marks would be movement of molecules or movement of particles from high to low concentration. That's the second mark. Okay, part B, it says give a definition of active transport. So active transport is against the concentration gradient, so it's from low to high. So I've included a diagram here. So again, it's going from a low concentration to a high concentration, and it requires energy. So you can think about it as pushing a boulder up a hill. So if diffusion is pushing a boulder down the hill, active transport will be pushing a boulder up a hill. It requires ATP, which is the energy currency in our cells. And not only that, but it also requires these protein carrier or carrier proteins. So let's write that here, and let me just remove this. Okay. It is the movement of molecules from low to high concentration using energy in the form of ATP. So ATP is the energy currency in our cells. This requires a carrier protein. So let's go over the marks. We've got movement of molecules from low to high. That's 1 mark. Using energy, that's the 2nd mark. ATP is the 3rd mark. And making reference to the carrier protein, that's the 4th mark. Okay, let's move on to part C. Give a definition of osmosis. So osmosis is the movement of water. Some people also call it the diffusion of water as well. So it goes from a region of high water potential to a region of low water potential. Actually, let me include a diagram here. So here we have a diagram. So osmosis can only occur through a partially permeable membrane that doesn't allow for the larger solute molecules to pass through and would allow for water molecules to pass through. So it is the movement of water. So here you would have low water potential. Here you would have high water potential, and the water is going to move from here to here. Okay, let's remove all of this. Let me erase this. Okay, so we would say it is the movement of water from high to low water potential through a partially permeable membrane. So the marks are Oh, where did the toolbar go? Okay, so the marks are movement of water, that's one mark. The other mark is high to low water potential. And the third mark is through a partially permeable membrane. Okay, let's move on to question 3. The diagram shows some of the components of blood. Part A, it says complete the table by giving the name of the component and its function. So let's do part A. So they've already done A for you, and it's a red blood cell because of the biconcave shape here. And B is a white blood cell. Specifically, it is a phagocyte, and phagocytes are responsible for engulfing pathogens or engulfing bacteria. We know it is a phagocyte because of this lobed nucleus that you see here. Let me show you a diagram of a lymphocyte. Hold on. So we can see here that this over here is a phagocyte. This is a phagocyte. Now these two over here, these are lymphocytes, and lymphocytes are responsible for producing antibodies, which could then bind to the antigens on the pathogen. Okay, let me just remove this and delete this. Okay, and then lastly, part C, that would be the platelets. And the platelets are responsible for blood clotting. Okay, part B1, describe the process of blood clotting. Okay, so I've included a flow chart for how blood clotting occurs. So platelets they're not actually cells, they're fragments of cells, they're cell fragments, and they would release an enzyme, which you don't need to know for your syllabus, but this enzyme would convert prothrombin to thrombin. So prothrombin is a soluble protein that is inside of the plasma, and then the thrombin will then convert soluble fibrinogen, so this is soluble, into insoluble fibrin. And that can form this nice meshwork that you see here, which can trap the red blood cells. Okay, so let me just remove this and write this down. So we can say that platelets release enzymes that causes prothrombin to be converted to thrombin. And then thrombin converts fibrinogen to fibrin, which enables it to form a meshwork that can trap red blood cells. Okay, let's go through the marks for this. Okay, so we've got platelets releases, that's one mark. Enzyme, that's another mark. That causes prothrombin to get converted to thrombin. Oops. That is a third mark. And the fourth mark would be fibrinogen to fibrin. And the fifth mark would be meshwork is formed that can trap the red blood cells. So that is the 1, 2, 3, 4, 5, 6 marks. Okay, let's move on to part 2. So it says, explain why it is important for blood to clot. So It's a 3-mark question. We can talk about how blood clotting is important for preventing things from coming in and from preventing things from leaving. Specifically, what we want to prevent from coming in are pathogens, because if pathogens come in, then that would cause infection, right? So it prevents pathogens from entering and causing infections. It also prevents blood loss or excessive blood loss. Excessive blood loss. So it prevents the entry of something and prevents the loss of something. Okay, let me just go over the marks. So 1 mark for prevents pathogens from entering, that's 1 mark. Causing infections, that's a 2nd mark. Prevents excessive blood loss, that's a 3rd mark. Okay, let's move on to question 4. The diagram shows the alimentary canal. Okay, let's scroll down. It says identify the areas of the alimentary canal using labeled lines and the correct letters. Label with an X the area where the starch digestion begins. So that would be the mouth. So I'm just going to label the mouth. So your mouth contains salivary glands, and the salivary glands secrete amylase, and amylase is the enzyme that is responsible for breaking down starch to maltose. Next one, part 2, label with a Y the area that has a pH of 2, so that is acidic, and the only part of the alimentary canal that is acidic would be the stomach. So this would be Y. Third part, label with a Z the area where both lipid and protein digesting enzymes are produced. So that would be the pancreas. So this is the pancreas here. So we will label that with a Z. So the enzyme that's responsible for digesting lipid is referred to as lipase or lipase. And the enzyme that's responsible for digesting proteins are referred to as protease. Okay, let's move on to part B. Some people have the condition known as celiac disease. This results in the villi being flattened. Explain why some people with untreated celiac disease will eventually develop osteoporosis. So osteoporosis is when the bone density decreases, and so that leads to the bones becoming more weaker. And the nutrient that is responsible for the development of our bones are calcium, so calcium ions. Right, and so if a person has celiac disease, it says that their villi is being flattened. Let me just include a diagram of the villi. So I've included a diagram here. This is what the normal villi would look like, and you can see your villi is basically these finger-like projections, and that would increase surface area for absorption of these nutrients. However, if a person has celiac disease, their villi gets damaged, it gets flattened, and so that reduces the surface area for which you can absorb the nutrients, which means in this case, it reduces the ability of the small intestines to absorb calcium. So let's write that down. Okay, so we can say when the villi are flattened, this decreases the surface area for absorption, which means I want to say less calcium is absorbed into the body. And then we can say required for bone development. So if the person does not receive enough calcium, it would cause their bone density to decrease. Okay, let's go over the marks. So 1 mark for decreases the surface area. Another mark for less calcium is absorbed. In the marking scheme, it says unable to absorb calcium. We can say calcium is required for bone development, for the bones. So that's 3 marks so far. And lastly, it would cause their bone density to decrease. Okay. Let's move on to the next part. Part C, it says another condition of the alimentary canal is colon cancer. This is often treated by the removal of the large intestine. Explain why some people have this treatment often suffer from dehydration. So if a person has their large intestine removed and that causes dehydration, then that means that the large intestine must be responsible for the absorption of water. So let's write that down. So the large intestine is responsible for the absorption of water, which means that when it is removed, not enough water is absorbed. Okay, let's move on to question 5. We'll cover this in the next video.